The overall goal of these labeling procedures is to image and quantify stable, dynamic, and nascent microtubules in the developing zebrafish embryo.
This method can help answer key questions in the field of developmental neurobiology, such as how microtubules contribute to the establishment of cell polarity.
The main advantage of this technique is that it optimizes the detection and quantification of distinct microtubule populations in situ.
Generally, individuals new to this method will struggle because of the difficulties in maintaining microtubule integrity.
After decorionating staged zebrafish embryos according to the text protocol, transfer them to 1.5 milliliter centrifuge tubes, removing as much medium as possible. Add 1 milliliter of 4% PFA in microtubule assembly buffer, or MAB, to fix the embryos at 28.5 degrees Celsius for 5 minutes. Then use a pipette to aspirate the fixative and replace it with 1 milliliter of fresh fixative. Incubate the samples on a rocker at room temperature for 3 hours. Aspirate the fixative and add 1 milliliter of TBS and P40 buffer. Gently agitate the embryos on a rocker for 5 minutes. Repeat the wash 2 more times. Then, after replacing the buffer with 1 milliliter of fresh TBS and P40, store the embryos at 4 degrees Celsius for no more than 7 days. After de-yolking and embedding embryos in agarose according to the text